Okay, good morning everyone, and welcome to the 2017 Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present Mr. Conrad Wilson. Many of you will be familiar with Conrad. As a teenager, he has been a participant in the Encana Badlands Science Camp, and then later returned to be a camp counselor for the past 3 years. Conrad is originally from Vancouver, BC, and has been passionate about paleontology ever since his childhood. He went to the University of Calgary to pursue a bachelor's degree in biological sciences. For his senior undergrad thesis, Conrad described a new type of primitive ray-finned fish, or Actinopterygian, from the Early Carboniferous of Nova Scotia. After successfully completing his bachelor's degree last year, Conrad has applied for graduate school and will be pursuing a PhD later this fall. Despite having worked at the Royal Tyrrell Museum for several years, Conrad's main research interest is not in dinosaurs, but fishes. So it shows that sometimes we fail. So in his presentation today, Conrad will discuss how the research he has conducted in the context of his senior undergrad thesis sheds light on the early evolution of ray-finned fishes. So without further delay, I present you Mr. Conrad Wilson.
Thank you for the kind introduction, Francois. I've admired this stage and this institution for a long time, and it's really an honor to be here. So thank you all for coming, and good morning. Today, I'll be presenting most generally on the topic of mass extinctions and diversity patterns in life history, the history of vertebrate life on Earth. And I will also present some of my undergraduate thesis work in describing a new species of fish and some of its implications for mass extinctions and the history of vertebrate diversity on Earth. At this point, I think it would be helpful, at least for me, if you were to think of a vertebrate, any particular vertebrate, and hold it in the back of your mind. Come back to it later. But now I'd like to talk about the basic classification of vertebrate life. Almost all vertebrates today are gnathostomes. They're jawed vertebrates. There are a couple of lineages that are not jawed, the hagfish and the lamprey. But the near totality of vertebrate life can be described as the chondrichthyans, the cartilaginous fishes, or the osteichthyans, the bony fishes. And the cartilaginous fishes include, well, sharks, rays, and their kin. And the osteichthyans over here include two major radiations, the actinopterygians, the ray-finned fishes, and the sarcopterygians, the lobe-finned fishes. And there's a lot of diversity within these groups. We have the actinopterygians up here, the chondrichthyans back here. Looking at the lobe-finned fish, they range from everything really from lungfish up to reptiles and mammals. Pretty much everything that you think about when you think of land animals is a tetrapod. It's a tetrapod, it's a lobe-finned fish, and it's a bony fish under the classification scheme. So I will argue that the museum has worked because the museum is interested in certain types of fish, and I'm interested in certain types of fish. So what did everyone think of when they thought of a vertebrate? Do we have any people think of mammals? Anyone think of a mammal? Yeah? Some dinosaurs maybe? Yeah? Reptiles, birds, that sort of thing? Anyone think of a fish? Anyone? Well, that's not surprising because we like tetrapods. We like animals that are on land. We like things that are like us. We like things that we can see every day. But if you were all picked from a list of all modern vertebrate species, probably more than half of you would have chosen a ray-finned fish. The ray-finned fish form more than 50% of all known vertebrate species alive today. The rest of the vertebrate tree is made up largely of tetrapods. Mammals only form about 7%. So the ray-finned fish is not only a large taxonomic grouping, but it's an incredibly diverse one. I've selected a few images here. I've got a sturgeon, a seahorse. There's some other crazy fish like anglerfish that live on the ocean floor. Really a lot of diversity here. But fish, this sort of fish at least, aren't particularly glamorous. They don't receive a lot of study, especially fossil fish. So our understanding of the early evolution of this group is, at this point, Since the early days of evolutionary study and just evolutionary thought, there have been a few different classification schemes for the ray-finned fish. They were as a group fairly early on. But with the advent of modern mathematical-based and phylogenetic, cladistic approaches to the study of evolutionary relationships, a lot of the old groups have broken down and discarded. You won't hear much talk about the Crucitorigians or the Palaeoniscoids. Palaeoniscoids are a garbage bin taxa, and it's been thrown out. But the question is where its former members fall into place. The point of these figures is not to dwell over the details of the clades, but to show that in the last decade, there's been a lot of work on ray-finned fish phylogeny, and our basic understanding of the early branches of the Actinopterygian tree is quite poor. The branches move all the time. We don't have a consensus of where these things fall. And the problem is not driven by failure in methodology or worker failure. It's simply a lack of data. Most early ray-finned fish exist as flattened smears on rock, so it's really hard to extract 3D data from them. Furthermore, a lot of these early taxa haven't been revisited. Taxa described in the early 1900s have not been revisited or reanalyzed with modern techniques. So there's a lot of information out there that could be helpful for our phylogenies, and there's been a big push recently to get a better understanding of the ray-finned tree at its early levels, but a lot of work still needs to be done. So I described the basic pattern of vertebrate diversity earlier on, with the ray-finned fishes really forming the backbone of vertebrate life. But this pattern hasn't always been around. And so the study of the early evolution of the ray-finned fishes is sort of tied up with the origin of the diversity, the related topics. And an area of research where paleontologists often turn when they're interested in origins of diversity and origins of groups is the study of mass extinctions. Mass extinctions can provide some pretty satisfying answers to some basic questions. But as our understanding of these phenomena has increased, we're finding that we need to apply more and more rigorous methodology to the study. So mass extinctions are a relatively new field in paleontology. Topics such as development and anatomy and genetics have received detailed study for 100 years. But the first reference to the word mass extinction in the scientific literature doesn't show up until the 1950s. And prior to then, there really wasn't good evidence for mass extinction in the vertebrate record. There was a lot of work needed to be done to even figure out that mass extinctions were a thing. In Darwin's time, many paleontologists were arguing against a worldview that relied on a single cataclysm to produce the entire vertebrate fossil record, the entire pattern of diversity seen today. They were arguing against that. Darwin and his contemporaries were well aware that things vanish rapidly in the fossil record. They knew that ammonites disappeared at the end of the Cretaceous, and trilobites disappeared quite rapidly at the end of the Permian. But from what they knew about the fossil record, that it was incomplete and sporadic, it seemed more likely that the incompleteness of the fossil record was obscuring a more gradual process of extinction. We were simply looking at one point where trilobites and ammonites were diverse, and then looking at again at a different point where they weren't. The intervening level wasn't preserved. So in 1952, when Norman Newell put out an appeal for workers to provide him with information about the diversity of life at different points in Earth's history, essentially he produced a list of what lived when, some interesting trends emerged. And this was reinforced by J.J. Sapkowski and David Raup's work in the late 1970s and early 1980s, which identified several points in the history of life on Earth where diversity dropped rapidly. They identified 5 major mass extinction events, what they call mass extinction events in Earth history. Some of us will be familiar with them. They are the End Ordovician, End Devonian, End Permian, End Triassic, and End Cretaceous, the big 5. And so working from these mass extinctions, they produced a definition for mass extinction, which I've provided here. Basically, for an event to qualify as mass extinction, a lot of different things in a lot of different places around the world have to go extinct in a relatively short period of time. It's rapid, it's global. But this definition is quite relative. You'll note the words substantial increase, which aren't defined. More than one geographically widespread, which is not defined. And relatively short, which is not defined. So different workers will, with different data, produce different interpretations of the history of mass extinctions. Sapkowski and Raup identified 5 with their data. Some have identified as many as 61. David Bambach, in his work on the subject, identifies 18 based off of a slightly different set of criteria. He includes the requirement that faunas change after mass extinctions. So there's 18 mass extinctions here. I find this a bit problematic, as that's many more mass extinctions than I have fingers on either hand. I can't keep track of them. But his analysis is based in data and good science. So at this point, I'd like to, for my further discussion later on, talk about the Sapkowski definition of mass extinction in a little bit more depth. I also present some challenges to our understanding of mass extinction, and I think this will be helpful when we discuss vertebrate diversity and ray-finned fish evolution later on. So the first major mass extinction is that it be geographically widespread and taxonomically widespread. It affect more than one major group. Perhaps our most famous mass extinction, the Young Cretaceous, qualifies as a mass extinction not because the dinosaurs go extinct, but because dinosaurs are lost alongside many other diverse vertebrate and invertebrate groups. The mosasaurs, the ammonites, and many different kinds of foraminifera go extinct over the Young Cretaceous, and that's what qualifies it as a mass extinction. The other major requirement is that mass extinction be the product of an increase in lineage termination, or an increase in the extinction rate, the rate at which things are going extinct over time. This is important for 2 reasons. Mass extinction is a global, ongoing, continuous process that's quite normal in life. Things go extinct over time. And the rate of extinction may seem arbitrarily high to us. We're concerned about extinction of life today, not because some things are going extinct, but because they are going extinct very rapidly, more rapidly than one would expect based on the fossil record and based on our understanding of the history of life on Earth. As an example, looking at the Devonian, the mean rate of extinction for non-peak extinction intervals over Devonian substages. So what I'm saying is the rate at which things go extinct when there isn't a mass extinction on and over a relatively short period of geological time is between 18 and 19%. So over these short intervals, 18% of species are being lost. And this is normal. Mass extinction has to be significantly higher than that. And 18 to 19% seems like a relatively high number to us. But it's geological time. The other factor to consider is the rate of origination, the rate at which new species and new groups appear in the fossil record. Under normal circumstances, origination rate tends to match the extinction rate. Those taxa that are lost are replaced by new taxa. However, if extinction rate exceeds the origination rate, then there will be a depletion of diversity. But this does not necessarily mean that there's been a mass extinction present. If extinction rate rises because of some global event or some process within the fauna itself and exceeds origination rate, then there will be a decrease in diversity, and we can say that there's been some sort of extinction event. Whether or not it's a mass extinction will depend on the other criteria. But if origination rate drops, which it has, below that of extinction rate, without extinction rate increasing, then there will also be a depletion of diversity. But it's not an extinction-driven process. It's quite different from a mass extinction. A good parallel for this is what's happening in say, many Western societies today. We can use this analogy. If we swap birth with origination and death with extinction, population size, or at least the incumbent population size, is decreasing. But it's not driven by an increase in death rate. There's been no plague, no global catastrophe. Instead, the decrease in population size is driven by a decrease in birth rate with economics and emancipation. So one has to consider the origination rate as well as the extinction rate when talking about mass extinctions, and this is not something that's been done until recently. But the real problem with our understanding of mass extinction is the issue of timing. The Sapkowski definition requires that mass extinction occur in a relatively short interval of geologic time. But the incompleteness of the fossil record and its sporadic nature means that timing these things is really quite difficult. Certainly, the incompleteness of the fossil record affects our estimation of extinction, and it affects our estimation of origination, but not in the same way or to the same degree that it affects our estimation of timing. I'm sure we'll all agree that it is quite impossible for an organism to continue leaving fossils behind after it has already gone extinct. If it's leaving fossils, it is not extinct. But it's quite easy for a taxon or an organism to stop leaving fossils behind before it has gone extinct. It's just not being preserved. This effect means that the last occurrence of a taxon in the fossil record can be significantly before its actual extinction. Behold the coelacanth. This was once thought to have been a casualty of the end Cretaceous mass extinction, as its last fossil record is in the late Cretaceous. That was until it showed up in a fish market in Madagascar in 1938, 65 million years later. Bit of a surprise. So the last record of a group or a taxon occurs well before its actual true extinction. The true extinctions are stars, and the last fossil occurrences are circles here, over any interval of time. So the casualties of mass extinction are strewn backwards through time. And the losses seem to occur over a much longer period than they actually did. So mass extinctions can look more subtle and more gradual than they really are. The coelacanth is really an exception to this. Most taxa that leave their last fossils before a mass extinction event go extinct in that mass extinction event. They don't have to, but we can model the probability of that happening in the distribution of taxa through time. The coelacanth really should be considered a casualty of the end Cretaceous mass extinction, probably anyways. It's what we might call a dead clade walking. It's hanging on, it's there. It has some stable populations, but they're limited in space and they're limited in ecological role. The coelacanth belongs to a once diverse lineage. It has not rediversified. There's only a couple of types of coelacanth when there used to be many, many, many different types. A more recently recognized and profound problem in our understanding of mass extinction events is the role that stratigraphy plays in determining the signal of mass extinction. Remember, we're not looking directly at the extinction of animals. We're looking at their last occurrence. And a lot of our understanding of mass extinction derives from the marine fossil record. Marine organisms are preserved more readily. There are many more of them. And there's a lot of invertebrates, hard-bodied invertebrates that are preserved. So we get a much more continuous, much more diverse view of the history of life on Earth using the marine record. Sapkowski and Raup produced their analysis of mass extinction almost exclusively from the marine record. If you add the terrestrial record in there, it doesn't really change much. So especially in marine settings, there are different environments. And different environments occur in different places in the rock record. So if in this imaginary stratigraphical column produced by a computer simulation, different environments are preserved. Here we preserve deep water environments, again here, and again here. The transition between these, the transition between the high stands and low stand system tracks occurs here. So we get lots of different environments preserved. But we know from ecology that different organisms and different taxa occur in different environments. And especially in the marine record, they tend to be preserved near those environments. So deep water sediments preserve deep water organisms. But in a given stratigraphic column of rock, we're not getting the entire picture. Right? In this case, we're only getting a limited glimpse of the deep water environment in this particular location. Sea level is changing. The environment is moving. It persists along with its organisms and taxa, which go extinct gradually. They die off. It's just normal. But since we're looking only at a very narrow section of this deep water environment and a very limited time window, essentially all of the last occurrences occur in the same place. They are pulsed signals of last occurrence. These last occurrences, if we saw the entire picture and if we looked down dip from a given stratigraphic column, would be spread out. But in this case, where there's no mass extinction signal, we've got several significant pulses of last extinction, 50 to 100 times more than the extinction we see in the intervening intervals. So we can end up with a signal for a mass extinction when there isn't actually one present. When there is actually a mass extinction present, like in this figure, we have this black bar showing and the gray box showing sort of the extinction interval. We can look again at the deep water environments, which are being preserved here and here, only to a limited extent. These deep water environments occur considerably before and below the actual level of mass extinction. And again, they're sort of collecting all of the last occurrences. Our extinction is here. Our biggest signals of last occurrences are here and here. So the mass extinction is showing up considerably before the actual extinction event. This didn't used to be such a problem because the methods used to date the actual mass extinction were imprecise enough that the error range included those signals for the last occurrences. The error range was big enough that it didn't matter. But now, because our technology has improved and our dating methods have improved, we're ending up dating our mass extinctions in many cases before they actually occur, and we're getting multiple pulses. Holland reports a few cases where, by analyzing sections down dip and applying his statistical and model of mass extinction, we're able to essentially put together pulses of mass extinction. Extinction events that were thought to be pulsed are actually just one big extinction event. And there is the potential for some minor extinction events that we recognize to actually just be stratigraphically produced things. So when moving from a broad picture of mass extinction, just based off of last occurrences and taxa disappearing over a very broad window of time, to the sort of level of resolution that we're now demanding, one has to be very careful. One needs to do a lot of work and have a lot of data. So the end Cretaceous mass extinction event, which is probably the most famous mass extinction event, is really important. I don't think anyone's doubting that. It's a major transition. But it's really the exception to the rule about mass extinctions. The end Cretaceous mass extinction is the only mass extinction for which there is a massive individual pulse over a very short window of geologic time. Other extinctions are certainly mass extinctions, but they take much longer. The last occurrences occur over a longer period of time. And as well, it's the only one for which we can invoke reliably a cataclysm that instantly changes things, and with contra Darwin. So I'd like to apply this topic of mass extinctions to our understanding of the diversity patterns of life on Earth. If we were to roll back the tape to the Devonian and go up and expand the Devonian reef upstairs, we would see a lot of vertebrates with which we would be completely unfamiliar today. During the Devonian, the agnathans, this is a large paraphyletic, not a natural group, that includes basically any vertebrate without a jaw. And in the Devonian, there was a lot more than the hagfish and lampreys that we have today. These guys were armored, and they were relatively diverse. They were holding on from the Ordovician and Silurian. If we start looking at jawed vertebrates, these are the placoderms. They sit sort of at the base of the Osteichthyon tree. Here, placoderms would be somewhere down here. We see some taxa that we might be more familiar with, like Dunkleosteus. His skull is upstairs by the Devonian Reef. It's a large predator, can get to 20 feet long. If you were to look at the Devonian Reef more, you might notice in the back corner a little Ptyctodont placoderm. These guys are small and tend to feed on hard, or they seem to have fed on hard-bodied prey. And they're really quite convergent on the ratfish today, but only at a superficial level. The internal anatomy is a lot different, but it can be confusing. Here's a Renanin placoderm. It's flattened, and it looks a lot like a ray, even though it's completely different and completely armored. And then there's other placoderms which are just weird and wacky. Here's Bothriolipus, which is fairly well known. And not only the placoderms, there are things like acanthodians hanging out still in the Devonian. These are commonly known as the spiny sharks, but really they're more closely related to the bony fish than they are the chondrichthyans. Though this assignment is only recent and it's rather controversial, we don't have the best handle on what, we hadn't had the best handle on what acanthodians are. As well, there's large predatory sarcopterygians. These are the lobe-finned fishes. The group includes tetrapods, and certainly in the Devonian, tetrapods are beginning to access land. The amphibians have diverged from the rest of the sarcopterygians. But the main axis of sarcopterygian diversity is along these large aquatic fishes. Go forward a few tens of millions of years, and one encounters the Carboniferous fauna. It'd still be unfamiliar, but it's a lot more familiar than the Devonian fauna. Instead of placoderms, we have many different types of ray-finned fish. They're different from the ray-finned fish today, but they are still ray-finned fish. You also have a lot more sharks. And tetrapods are really starting to make an impact in the vertebrate fossil record on land. So what's going on between the Carboniferous and the Devonian? If you recall from Sapkowski's record of vertebrate life on Earth, the end of the Devonian is marked by a significant divot in the trajectory of vertebrate life. Sapkowski identified this as a single mass extinction, the end Devonian mass extinction. But more recent work tends to consider it as the end Devonian biodiversity crisis. This divot at higher resolutions of time seems to be more the product of two mass extinction events. The Kellwasser, which is on the left here, it occurs about 374 million years ago in the Devonian. It's at the boundary between the Franian and Famennian stages. It's followed relatively quickly by the Hangenberg extinction, 359 million years ago, or thereabouts. And this occurs at the very end of the Devonian and the immediate onset of the Carboniferous. It's the border between the Famennian and the Ternesian. So a lot of previous work has focused on the Kellwasser. It's not up there with the Cretaceous Paleogene extinction or the Permian, for sure, but it's received a lot of attention. It's a big signal, especially in the invertebrate fossil record. About 60% of marine taxa go extinct over the Kellwasser extinction. But most recently, work by Lawrence Sallin and others has focused on the Hangenberg extinction, specifically reference to the vertebrate tree of life. Over the Kellwasser, vertebrates aren't doing particularly badly. About 19% of vertebrate taxa are lost across the Kellwasser. If you recall, the background rate of extinction for the Devonian is about 18 or 19%. There's not a big difference. I'm not exactly comparing apples to apples. It's worth noting here we're talking about genus loss over a short interval of time and species lost over a somewhat longer period of time. But you can make a broad comparison. The Kellwasser level of extinction is not that much higher. The Hangenberg, though, is much worse for vertebrates. 34% of vertebrate taxa go extinct across the Hangenberg extinction. For those familiar with other mass extinctions like the end Permian and end Cretaceous, this is not a particularly high level of genus loss. We're accustomed to figures like 90% for the end of the Permian. The Hangenberg becomes more profound, though, when one looks at the level of higher clades, higher groupings of taxa, like the different types of placoderm or the different types of agnathan. What we find is that 44% of higher level groupings disappear over the Hangenberg extinction. This is not, the word higher level class or clade is not defined per se in the original work. There's a little bit of leeway, but if we're using an older taxonomic scheme, we're looking at the level of families and classes, really. We can see the effects of this here. The Kellwasser extinction occurs at the border between the Franian and the Flaminian. The Hangenberg extinction occurs between the Flaminian and the Tournaisian. There's a big drop in many different vertebrate groups over the Hangenberg, and not so much of a drop over the Franian and Flaminian. We can look at this figure in more detail and differently. Here's a chronological listing of all essentially known fossil-producing vertebrate localities for the Devonian and Carboniferous. We have our placoderms in green here, our sarcopterygians in purple, our ray fin fishes in light blue here, and we've got some things like the acanthodians and the climatids and all these related taxa in a darker blue at the bottom. The acanthodians are this light red here. So over the Kellwasser event, well, some placoderms, some acanthodians, some agnathans, some sarcopterygians go extinct. But they're immediately replaced by more placoderms, more sarcopterygians, and more acanthodians. The colors don't change. But over the Hangenberg event, at the end of the Devonian, the colors change a lot. We now have actinauts in light blue, sharks, chondrichthyans, in this lighter red here. The placoderms vanish. They go extinct completely. Sarcopterygians, in their purple, become marginalized and relatively few in species and number. And the acanthodians become a dead clade walking, like the coelacanth. They persist much later into the Phanerozoic, but they're really only known from one body plan and one real taxon. They're not doing too well. Another way to look at this chart is a canonical correspondence analysis of the end Devonian mass extinction, or the Hangenberg extinction. And this sort of thing is really attractive because these localities, they're the same localities that were shown here, these dots, are not plotted by age or by environment. They're not sorted at all. Instead, they sort based off of their similarity to one another in terms of faunal composition. If a site has the same fauna as another site, it will plot relatively close to that site. But if it's got a completely different fauna, if the site is unrelated, it'll plot completely differently. So you can see the two distributions. And the axes are really quite arbitrary. So looking at the earliest Devonian stage, the Jevetian, as well as the Franian and Flaminian, these are the intervals, the Franian and Flaminian, over which the Kellwasser extinction occurs. They overlap. The different trajectories of the points, they're the same. There's not much change. The Carboniferous faunas and localities plot together as well. The Serpicovian, Visaean, and Tournaisian sites are all grouped together over here. You can also group these by environment with a similar result. So the Kellwasser isn't producing a difference, but the Hangenberg extinction at the end of the Devonian is. I'll say a brief word about cause here. Compared to some more famous mass extinctions, like the end Cretaceous and end Permian, there hasn't been a lot of work done on the Hangenberg. It's not really been recognized as important until most recently. There's been hypotheses out there ever since the end Cretaceous impact. Asteroid impacts have become popular. That's been proposed with limited success. The most widely accepted view is that the Hangenberg extinction is caused by glaciation on a scale similar to that of the Pleistocene. It's affecting marine environments and disturbing the equilibrium there. But the important thing for my discussion in this talk is not the cause, but rather what's happening with the faunas. The Hagenberg extinction is founding the pattern of vertebrate diversity seen today. Actinauts become dominant, and they're accompanied by tetrapods here in orange and chondrichthyans. And the actinauts never let go. They are the most common form of vertebrate life today. They've changed. They've gone through many different evolutionary radiations, but they are still the backbone of vertebrate life on Earth. So surely, given that our understanding of actinaut evolution is poor, and we know that the Hagenberg extinction is having a tremendous effect on their diversity, surely we must be looking at the interval immediately after the Hagenberg extinction, the Tornasian stage, for some of these answers. But we're not. We haven't been. I'll mention this here. We have the Hagenberg extinction here about 360 million years ago. And this is a chart of sort of the basic divergences for some ray-finned fish clades. These sort of charts where we estimate the age at which these groups split are produced based off of a backwards estimate of genetic rates of change. We look at two taxa that are related today, figure out how genetically distant they are, and sort of extrapolate backwards. So we assume a constant average rate of genetic change. And so based on the data set used, what sort of DNA, what sort of calibration, you get up with fairly widely different results. There's a lot of variance. You get some dates estimating the actinoteri as diverging 350 million years ago, others closer to 400. There's a lot of change and a lot of variance. But one of the things to consider is that genetic calibrations, these molecular clocks, can't account for mass extinctions. Because they're using an average rate, they can't account for potentially higher rates of genetic change and diversification following a mass extinction event. They'll produce a date older than the actual divergence if things are speeding up after a mass extinction. I won't say anything more than that it is interesting that a lot of these basic divergences are occurring around the Hagenberg. This may be a false signal, and maybe not. It's something to look into. So back to what I was going to talk about before I remember this slide, the Tornasian interval. We know from our understanding of vertebrate diversity and a basic look at the molecular clock that the Tornasian should be an important interval for the ray-finned fish evolution. We should be looking about 350 to 340 million years ago for some answers. But this has been a huge problem in vertebrates. The end of the Carboniferous, and the earliest Carboniferous, rather, is an interval known as Roemer's Gap. The influential paleontologist Alfred Roemer first identified this in 1956, and it's been a problem since then. We've got this very fossil-poor interval, at least for the vertebrate record. And it's really a major problem. Recent work has reduced it to 15 million years. When Alfred Roemer first described it, it spanned the first 30 million years of the Carboniferous. Now it's only 15, an interval that equates really to the Tornasian stage. But 15 million years is still a really long time. If we go back to the end Cretaceous mass extinction and look at faunal change there, 15 million years takes us from dinosaurs, at least in the macropaunas, to some reasonably well-established mammalian communities. Whales are beginning to diverge at this point in Earth's history, 15 million years after the Cretaceous. So it's a big gap, and a significant one if we're trying to understand an important faunal transition. So what we know about the post-Hagenberg fauna is really limited. We know that it was probably smaller, that it had more actinopterygians, but there's a lot of uncertainty. In the original study identifying the Hagenberg event as a significant extinction in the vertebrate record, only 5 of the 66 localities considered were Tornasian. 5 of the 66, not very many. Same for similar numbers for this analysis of size change across the Hagenberg extinction. There's room for error if we're only considering 5 localities. But the idea that the post-Hagenberg fauna was smaller fits generally with our understanding of mass extinctions. There seems to have been a change in feeding strategies, at least among those vertebrates who are feeding on hard-bodied organisms. These are called durophagous predators. They seem to be changing their feeding strategies and becoming more numerous after the Hangenberg. But this is really all we know. The problem with Romer's gap is huge in our understanding of the Hangenberg extinction, especially when one considers the amount of data one needs to work accurately with mass extinctions. Now, there's been a few hypotheses proposed for the cause of Romer's gap. There's even been debate on whether it's a real sort of biological phenomenon produced by a geological or biological process, or whether it's simply an artificial modern-day collection failure. It was proclaimed that Romer's gap was the product of low atmospheric oxygen for these 15 million years. But it's not actually that low. New data has suggested that it's not that low. And it's never really been clear how atmospheric oxygen would only affect terrestrial environments and produce this change in this distinctness in the tetrapod faunas, but not in aquatic environments. Since the identification of the Hangenberg extinction as a mass extinction, it's been also suggested that Romer's gap might just be a post-extinction trough in diversity. Things are recovering. And this gels well with what we know about mass extinctions. It's probably accurate to an extent. But it turns out that when we actually go looking for Tournaisian localities, when we investigate Romer's gap, we get a lot of diversity. Recent finds in Scotland have yielded diverse tetrapod, lobefin fish, other fish faunas, as well as diverse arthropods, including, and this discovery in Scotland includes a new, previously unrecognized radiation of durophagous sarcopterygians. In Canada, we can look at the site of Blue Beach, Nova Scotia, as a good case study for the sort of processes that are creating Romer's gap. We've known about Blue Beach in Nova Scotia for a long time. There's been collecting there, I think, since the 50s, but not continued collecting. Blue Beach is near Hansport, Nova Scotia, on the banks of the Avon River. And the Avon River sits in confluence with the Bay of Fundy and its record-breaking tides. The site is underwater for much of the day. Erosion is extreme. Fossils are mostly found on this beach here as loose, undifferentiated material, not much in situ. And anything that is not collected tends to be washed out to sea and destroyed. It's really not a place where you want to be collecting. But it's the only Tournaisian exposure for miles, or considerable distance. So with the establishment of the Blue Beach Fossil Museum recently, there's now been a base for continued collecting. And there's a lot more specimens and taxa coming out of Blue Beach. We're looking at some tetrapod fossils here. Anderson et al. Describe many in their most recent paper. But these are badly worn. They're not well preserved. And it's actually impossible to identify them down to genus or species. But we can be quite confident in identifying them to higher groups of tetrapods, like the colostiids, or the wachirids. And we get a surprising result when we do this. We see that the tetrapods at Horton Bluff are really a mix of typical Devonian and Carboniferous forms. There's Devonian forms there. They're mingling with the Carboniferous forms. There's mixing going on. So it looks like the tetrapods, as they survive the Hangenberg extinction, are actually surviving quite well. They're not making it across this extinction as uniform stragglers, but instead diverse lineages. But what of the ray-finned fish? And this is sort of where my undergraduate thesis work comes in. Two partially complete, 3D preserved ray-finned fish skulls have been recovered from Horton Bluff. I described one as part of my undergraduate research project. Here we have it in dorsal and ventral view, so we're looking at the top and bottom of the specimen. Looking at the skull roof, we're looking at the top now. We can identify the right parietal here and sort of base our understanding of the rest of the skull roof off of that. We have here a temporal flange that sort of sets the back of the skull roof in this animal, in this group. We have the right parietal here and a bunch of other bones here. This is sort of a superficial character, it's a superficial observation, but generally the pattern of the bones, if one accounts for the fracturing and displacement along the midline, is rectangular. They sort of match what we would see in a lot of early ray-finned fish, especially Devonian forms. If we look particularly closely at the skull roof, we see these ridges of hypermineralized tissue. This is likely enamel, the same material that you have on your teeth on the roof. It's forming a sort of ornamentation there, which is again reminiscent of some Devonian forms like Calgary or Rhinarius. Looking at the back of the skull, sort of the brain pan and the very back and bottom of the animal's head, we can see the parasphenoid. This is the base of the parasphenoid, rather, which forms a lot of the palate of fish. If the animal was complete, this would extend way up as sort of like a prong coming off. But we have the base, it's broad, it's triangular, it's very similar to something like Moitomasia or Rhinarius again, which are both Devonian forms. Crucially, I don't think it has ascending processes, which are somewhat diagnostic of later Carboniferous ray-finned fish. We see the lateral dorsal aortae here. These are the blood vessels that take oxygenated blood that has passed through the gills and through the brain back into the body. And these pass through the caudal region here, where the spinal cord and the notochord enter the skull. So because we had this sort of 3D preservation, we put it in a CT scanner. Here's the result. If you're used to working with CT data, this is not really the sort of thing you want to see. It turns out that the fossil is of a similar density to the rock around it, so you have a lot of difficulty distinguishing between the bone and rock of the specimen. You can see things like the skull roof here and the parasphenoid here. So I process this. I produce a 3D model. Here it is. All of these elements, like the base of the parasphenoid, in the actual model can be pulled apart. You can look at them separately. I've put them together here in the same color to avoid going into too much detail about the anatomy. And looking even further, you can extract information about the vasculature and what's going on with the blood vessels and the nerves inside the brain. Here's the vasculature. We have the internal carotids, which pass through the parasphenoid as they supply blood to the brain. If the specimen wasn't sheared here, this parasphenoid should be lining up with the lateral dorsal aorta if it wasn't broken. These would line up with the lateral dorsal aorta here as they pass through the skull. And the lateral dorsal aorta eventually merge. Again, this merge would be a lot cleaner if the specimen was complete into the dorsal aorta, which finally leaves and enters the body. The fact that the lateral dorsal aortae are present as grooves on the base of the skull is a typically Devonian characteristic. In later fishes, or ray-finned fishes, these spend more of their time within bone. They do spend some of their time within bone, they merge, but in later taxa, the entire thing would be covered by bone. So overall, this paints the picture of a relatively basal actinopterygian. It's got a lot more in common with Devonian forms than it does Carboniferous forms. And unsurprisingly, given that I coded the thing, the phylogenetic analysis sort of replicates this result. It comes out somewhere near here, which is with Moitomasia, which is a Devonian actinopterygian prior to the later Carboniferous radiation of fishes. So this carries with it a few implications. First off, just like the tetrapods, it seems that there's a lot of ray-finned survivors of the Hangenberg extinction as well. This is not to say that the two faunas are not distinct, the Carboniferous and Devonian faunas are not distinct at other levels. When you look at placoderms and stuff, there's a lot of change. But the change is occurring in the groups that go extinct, not the groups that are surviving. The actinauts pass through relatively unscathed. This new taxa adds another to the considerable number of lineages already known to pass through the Hangenberg extinction. And with the tetrapod data, we can really falsify hypotheses like the low oxygen one that require a big difference between the survivors of the Hangenberg extinction. And the last real implication is that when we're studying early ray-finned fish evolution and despairing about the lack of fossils in the Tournaisian, there is hope. Stuff can be found if you look, and in some cases it's 3D. Application of the technologies like CT scanning to new taxa and older taxa can give us a much better understanding of the anatomy and the evolution of the ray-finned fishes, and potentially help solve an enduring and significant problem in vertebrate evolution. I've got a lot of people to thank for this. I'd particularly like to acknowledge my undergraduate supervisors, Jason Anderson and Jessica Theodore, my lab mates, Jason Pardo and Matt, as well as Matt Friedman for his helpful criticism and advice. And thank you very much for your attention.